Hello everyone, welcome and thank you for attending our fourth Vitalograph webinar in the series on considerations for clinical research. Today's topic is clinical research considerations for lung function assessments in ALS studies. My name is Grant Soman and I'm the Vice President of Clinical Services at Vitalograph and will be the moderator for today's webinar. Before I introduce today's speaker, I've just got a few housekeeping items. You will be muted during the talk. But there will be time at the end for a Q&A session. So we do invite you to use the chat function to type any questions you may want to ask our speaker. Also, there will be a Q&A log available on our website after the webinar. This webinar will be recorded and we'll be in contact with you on how you get access to this recording. This live webinar is accredited for 1 hour's CRCE credit by the AARC and it's available to members. It is a requirement that the attendees complete a feedback form, which we'll send out at the end of the webinar so we can provide AARC with evidence of your attendance. Here at Vitalograph, we will also be running more webinars, so please do look out for these in the near future. So that's the housekeeping items done. So I'd like to introduce our guest speaker for today. Chris McDermott is Professor of Translational Neurology at the University of Sheffield and Team Lead for Neuroscience at the NIHR Sheffield Biomedical Research Centre. Chris was awarded a prestigious NIHR research professorship to improve care for people living with motor neuron disease. Chris chairs the National Motor Neuron Disease Clinical Studies Group and is the co-director of the new MND National Institute. He works to harness data to optimize the timing of care interventions and develop decision aids to help patients make informed choices about the care they receive, develop remote monitoring to improve clinical trial outcome measures, widen access to research and improve patient services. His research interests center on improving clinical trial design, device development and improving symptomatic care. We are very excited and pleased to have Chris here today to talk to us on clinical research considerations for lung function assessment in ALS studies. So with that introduction, I'll pass this over to you now, Chris.
Thanks very much, Grant. Okay, so for the next 45 minutes, we're going to talk about motor neuron disease, ALS, and particularly the clinical trial landscape. There are many things within the field of motor neuron disease that are changing. So having updated us all and give us some fast facts about motor neuron disease so we're all on the same page. I'll then talk about how things are changing, what the current treatments are, and then reflect on how we might use care and research, how we might use technology for care and research. So first of all, motor neuron disease is commoner than people think. There's a 1 in 300 lifetime risk. In the UK, for instance, there are 5,000 people at any one time living with motor neuron disease, but that population, because of the dire prognosis of motor neuron disease, often is changing. 50% of people with motor neuron disease will die within 2 years, and there is no cure. It's an awful disease where people are robbed progressively of the ability to speak, to chew, to swallow, to move, to use their arms, and eventually to breathe. When I'm talking to patients, we talk about the connection between the brain and the muscles. So I liken the motor nerves to wires, and in motor neuron disease, that connection, those motor nerves or those wires are degenerating, and eventually paralysis ensues. This is a cartoon of those wires, and if we look at what's going on in a sick motor neuron, there's a lot of things going on that we don't want to see. There's inflammation, there's apoptosis, mitochondrial dysfunction, excitotoxicity, many different pathogenic pathways are activated in motor neuron disease. It's a complex disorder, and that's one of the reasons why we've struggled to find treatments. The barriers to progress have been this lack of understanding of the basic biology in motor neuron disease. Why are those motor nerves degenerating? What are all the different pathways involved? But also how we've been doing clinical trials, and that ranges from poor candidate selection, which in the past, because we didn't understand the disease so well, was often just trying things rather randomly because it hadn't been tried in motor neuron disease before, rather than there being a really strong scientific rationale. In terms of trial design, the inclusion criteria have not been perhaps as they should be. Outcome measures not ideal, and the whole setup and infrastructure around clinical trials has been inefficient. So I think these are some of the barriers, but we are beginning to make progress in all of these areas, and I'll highlight some of these. So in terms of our knowledge, major advances have been the discovery of TDP43, advances in genetics and biomarkers, and I will go into a little bit more detail on some of those. So the first thing is TDP43. At the top there in the little box, that's a motor nerve, a sick motor nerve. And you can see the brown blobs within the motor nerve there. They shouldn't be there. That's TDP43. For decades, we've been able to visualize that the sick motor neuron is chock full of inclusions, but we haven't known what the protein is. It's only in the last 10 years or so that that's been identified. And really, if you think about it, how could we ever think that we were going to be able to fix a problem if we didn't know what the key pathological hallmark was? What were these inclusions? Now that we know it's TDP43 and we begin to understand the function of TDP43, it's opened up a whole new area of research and potential targets for motor neuron disease therapeutics. One particular area is that of RNA metabolism, an area which really was of little interest, if any interest, prior to the discovery of TDP43. Now we recognize it as one of the key central pathological pathways within motor neuron disease. So a huge transformation in our understanding since the discovery of TDP43, and this is growing. In terms of genetics, so in the graph there, you can see the exponential increase in our understanding of the genetics of motor neuron disease, and this is work that has come out in a large part from the Project Mine collaboration. So approximately 10% of motor neuron disease is familial. The other 90% is sporadic, although even within those sporadic cases, we know that there are small effect gene changes that are contributing. Each of these genes that have been discovered, though, what they tell us is that that gene is important in motor neuron disease health and important in motor neuron disease when things are going wrong. And it's again about telling us information, potential targets, and a real understanding of motor neuron biology. So even though in these cases they may be rare families, and it might not be directly applicable immediately to the larger sporadic cases, the information that we learn about what is important in motor neuron disease in terms of motor neuron disease biology is really pushing the field and helping us introduce new targets. We now consider motor neuron disease to be a multi-step process. A number of things need to go wrong for someone to develop motor neuron disease, and there's been some mathematical modeling done, and it's been conceived that there are 7 steps to motor neuron disease that need to go. 7 different things need to have happened before someone gets motor neuron disease. In terms of what's happening in the genetic subtypes of motor neuron disease, these are a bit like if you've played the game Snakes and Ladders. They shoot people up the pathway. So rather than needing 7 steps, an aggressive SOD1 mutation might actually put a ladder up that only 1 or 2 additional things are needed, which may be an environmental factor or a polymorphism in another gene to get people up to the top and then develop motor neuron disease. So this explains some of the variability that we see. The other big development is the recognition that neurofilaments are a useful biomarker of neurodegeneration in motor neuron disease. Neurofilaments, it's an old story, and people have looked for correlations with neurofilament levels in the past, and the results have been unconvincing. What has changed is the technology now to be able to detect neurofilaments is much more precise, much more sensitive, and we are seeing robust evidence that neurofilament levels are elevated in motor neuron disease. That tends to be constant throughout the course of the disease, and as I will show some data, we see neurofilament levels lowering with treatment that has an effect on clinical outcomes. So this is really recent advancement, and I think will change the way we do clinical trials. So thinking about the management broadly of motor neuron disease, so a clinical trial, what sort of landscape is it occurring in terms of standard care? What does standard care look like? There are 3 broad pillars to the management of motor neuron disease. First of all, neuroprotective therapies, then symptom control. There are many symptoms that an individual with motor neuron disease is experiencing, and there are different levels of evidence of how we manage those symptoms, and some of those interventions themselves can have an effect on survival and also have an effect on some of the outcome measures that we're using in trials. So it's important to be aware of them. And then underpinning all that is a multidisciplinary approach to supportive care. And as I will show, that's also important to recognize because that can affect survival as well. So first of all, neuroprotection. Riluzole was the first agent to be shown to have some benefit in on survival, and so this is the initial data from 1996 showing around a 3-month survival advantage for those taking Riluzole. And Riluzole is pretty much standard care throughout the world, and most clinical trials would expect people to be on a stable dose of Riluzole, either on Riluzole or not on Riluzole at all. It's a standard entry requirement, and no change, hopefully, of Riluzole either on it or off it once they go into the trial. There are other neuroprotective agents, which I'm now going to come on to, but this is a much more complex situation. So we now have different regulatory bodies around the world approving newer neuroprotective agents based on different levels of evidence. So the first of these to highlight is edaravone. So this is the phase 3 data which took place in a selected cohort of ALS patients. So this is study 19, as it's referred to. This is the data on ALS FRS. So this is the ALS functional rating scale, and the R stands for revised. This is one of the most commonly used functional outcome measures in motor neuron disease. It is a questionnaire which relates to function, and the patient will complete it with the help of a healthcare practitioner or researcher. And in the phase 3 study of edaravone, there was a small but significant difference in the FRS score in favor of edaravone. This was intravenous edaravone, so this is quite a burdensome treatment, but it was licensed in some territories, but notably not licensed in Europe, certainly not widely in Europe. So we have a transatlantic divide over licensing here. In terms of how things are developing with edaravone, there is an oral equivalent, well, that we hope will be an oral equivalent, so that will reduce the burden of administration. And there is a phase 3 trial in the going on called ADORE, which is looking at daily dosing of oral edaravone. And it's useful to look at the design of the trial, because this is a particularly standard ALS trial design. So you can see the eligible participants. That is pretty typical of an ALS trial. So symptom onset within 2 years, SVC above 70% at entry, and change in ALS-FRS score pre coming into the trial, so that we're certain that people are progressing, and they're not going to be slow progressors, or not people that are going to be too fast in progression. So getting those people that are going to contribute data into the trial, 2-to-1 randomization, and the primary endpoint here being ALS-FRS, and then you can see the secondary endpoints, pulmonary function, amongst others. The other thing where there are territorial differences would be Amylyx 0035. So this is the phenylbutyrate and TUDCA combination. So the Centaur trial showed a significant difference in ALS-FRS in the randomized period, and then in the open label extension. Here you can see the randomized phase in the first part, and then it moves to open label extension, and there's a difference in those who received the Amylyx compound, the PB and torso, early on, compared to those who received it only at the start of the open label extension in terms of survival. So this was a given condition approval recently by the FDA, and also approved it in Canada. There is a phase 3 study now ongoing called Phoenix, which is nearing recruitment, which hopefully will provide the data that confirms the Centaur trial data. But at the moment, if you were starting another clinical trial, you may have the situation where edaravone, PB and TUDCA and riluzole would be standard care in the States, whereas only riluzole would be standard care in Europe. So there are differences that need to be taken into account. I'm going to talk about tofersen. So this is a Biogen study. I'm going to talk about the VALOR study. So tofersen is a therapy for SOD1 motor neuron disease. So motor neuron disease caused by SOD1 mutation, and this accounts for about 1% of all motor neuron disease. So it is relatively rare. And one of the nice things about these precision medicine approaches is you don't necessarily need to understand everything that's going on with the disease and all those complex pathways that I showed at the start. Here, the SOD1 gene is faulty, it produces a faulty mRNA, and that leads to faulty protein. We don't really fully understand how that then goes on to cause MND. So we know it's some sort of toxic gain of function, but the complete process has not been unraveled, it's fair to say. But in this case, it doesn't matter if we just stop the production of the faulty protein with its toxic gain of function. So this is the data from the paper earlier this year in the New England Journal of Medicine about tofersen. And tofersen is an antisense oligonucleotide. It needs to be given monthly via a lumbar puncture into the CSF, and it causes an enzymatic degradation, leading to a reduction in SOD1 levels. what is nice is that within this study, we're able to see target engagement. So you can measure the SOD1 levels. So just my lights are going off, which is not good. There we go. You can see the reduction in SOD1 levels. So in the VALA, you can see the placebo control arm in the dotted line. In the green line, it's reduced with the tofersen. When we get to the open label extension, when the placebo control switch on to tofersen, we then see that they're getting a reduction in their SOD1 levels. So we're seeing target engagement. Neurofilaments. So I talked about the developments in neurofilaments at the start, and here we have the placebo again in the dotted line compared to the people receiving tofersen in green. You can see a marked reduction in the neurofilament levels in the active treated group, and then when the placebo group go into the open label extension and start receiving active drug, the neurofilament levels come down as well. So this is really the first time we've seen this type of response. And we're seeing an effect on clinical function. So here the ALS-FRS score. So this is this questionnaire which talks about the daily functions that people are able to do. And we can see that the decline is slower in the treated group, and when people switch over to into the open label extension, we can see that those who received tofersen first, it continues to separate out. So the benefit of tofersen on the clinical function comes a little bit later, and so is only really being seen significantly in the open label extension phase. Similarly, respiratory function, we can see the separation in the lines, and when we get to the open label extension phase, the effect becomes more apparent. So what are the implications of the SOD1 work? So for the 10% with familial motor neuron disease, there are other promising gene trials underway now. So C9 is a gene which accounts for a large proportion of familial motor neuron disease. There's also trials in FUS-related motor neuron disease underway. So similar technologies being used for a precision medicine approach. But for the broader sporadic MND population, I think there are important implications of this work. So the neurofilament story that we're seeing here may enable faster clinical trials with biomarker readouts being used to give us more confidence earlier on about a asset, and using the ASO technology to influence key MND genes. So I showed all the different genes that we know to be involved in motor neuron disease, and our understanding of the MND biology, how it's evolving. We are beginning to target genes that can improve motor neuron disease health, or disease or genes which are deleterious and contributing to the degeneration, reducing their effect using ASO technology. And there are a number of studies coming online in the next year in the sporadic MND population using ASO technology. So other aspects of standard care, just to emphasize, multidisciplinary care. So it's important when we're choosing trial sites to understand what their standard of care is, because the multidisciplinary approach to MND ALS care improves survival. And what the data I'm showing here is looking at survival in a multidisciplinary clinic where the patient is seeing speech therapists, occupational therapists, physiotherapists, all these people working together, as opposed to just going to a general neurology clinic where they still might be seeing some of these other professionals, but it's not in a coordinated manner. And this survival is independent of whether someone gets riluzole, the feeding tube, or breathing support. So there's something else going on, whether it's related to the provision of hope. There are aspects of ALS that can be treated, and I'll touch on those in the next slide. Typically, MDTs are seeing patients every 2 to 3 months, so they're really keeping on top of every aspect of the disease for the individual and supporting them, helping them get the things that we know work, but also helping patients avoid the things that they might come across on the internet which don't work. So it's giving them proper guidance and advice and support. So symptomatic management is important in motor neuron disease, and some of these things do make a difference to survival. So the management of respiratory failure, I will use an example shortly, makes a big difference and probably has the biggest impact on survival, the magnitude of 7 to 8 months, really non-invasive ventilation. So it's important that when designing the clinical trials that although it's often an exclusion criteria going in to have non-invasive ventilation, many people are likely to start using non-invasive ventilation at a point within the clinical trial. So it's important to understand, I think, how that's being administered and delivered, because of the impact it has on survival. And then things such as oral secretions and management of oral secretions. If we take the ALSFRS, there's an item on the ALSFRS which asks patients about whether they have problems with oral secretions. Managing the oral secretions may improve that item. So you might get an improvement in the functional rating scale because of symptom management. So it's important that these things are accounted for or understood when trials are being designed. One thing which is important to consider is how people access care and trials. So in the UK, we have a number of M&D care centres, but you can see they're not everywhere, and patients have to travel significant distances to be able to access care and clinical trials and the survival benefit associated with these. One of the things that's beginning to develop within the M&D field is the use of telehealth, and so there are telehealth systems in a number of countries now, which all have their own flavour. So in the UK, we've developed the TIM, telehealth in motor neuron disease pathway, and this was designed with patients as a user-centered design approach to create something that patients wanted and could use. And we're recording proms are sent out to the patients, and they record the disease progress, and we're also capturing what's happening to patients, and we're recording ALSFRS, objective measures, medical device use, various symptoms that patients are experiencing. And we've explored with European colleagues in over a number, so in the Netherlands and Ireland, a survey to understand attitudes to using remote health technology, and largely people do accept this and are happy to self-monitor their health. They're happy with remote monitoring with somebody looking at the data to see how their health is progressing, their disease is progressing. Respiratory muscle strength was something that was valued in terms of home monitoring. In terms of overloading people with devices and apps, the acceptable limit for people was 3 devices, no more than that, and interacting with them around once a week was deemed acceptable. Of course, we are a spectrum with our attitudes towards these things, and 15% said that they wouldn't want any type of home monitoring, wouldn't want to do remote participation in the clinical trials, because they actually felt there was a burden then in their home of having this equipment and doing these things at home. There were also some who raised privacy and data issues. The service that we've set up, and the service is similar in other countries, so similar in the Netherlands, similar in Ireland, and others, I'm sure, is that the telehealth service links the patient and carers with the clinic, and enables in between those 3 monthly visits when people are being seen in clinic to have contact with the centre when needed, with the healthcare professional reviewing their data. And the things are evolving to include remote spirometry, keeping an eye on mobility with actigraphy, looking at detection of falls, so being able to intervene as things are happening rather than just finding out about things at a fixed clinic appointment. And developing things such as cognitive assessment within these remote platforms. So focusing on the respiratory aspects, in terms of standard care, I touched on the fact that this has the biggest impact of all the interventions that we have currently. A survival advantage of probably at least 7 months in the trial from 2006 by Stephen Burke, but I think most people accept it's probably a bit longer than that in the real world now, and how we're delivering respiratory support. And this would be just using the non-invasive ventilation overnight for at least 4 hours. It is a complex intervention though, and there are many factors that influence the quality of the ventilation delivered. And some of those are service related things and healthcare professional things, and some of those are patient related things. And we've designed a number of resources around this to help healthcare professionals ensure that they're delivering effective ventilation. So it's the NIV for M&D toolkit. And we've also developed with patients a resource called My Breathing, which helps patients right from the start when they might just be having respiratory screening, why they're having respiratory screening, why they might at some point need non-invasive ventilation, giving them information to support their decision, what is living with non-invasive ventilation like. And these two resources help support what is a complex intervention. In terms of management of motor neuron disease, there are a number of guidelines around in different territories. In the UK, we have National Institute for Clinical Excellence, and that has a guideline on motor neuron disease assessment and management, and part of that covers the respiratory assessments. So forced vital capacity or vital capacity, and there are various triggers for when one would consider referral for non-invasive ventilation. So the traditional monitoring in clinic is a 3-monthly VC or a SNP and a peak cough flow, looking at expiratory function. These are obviously challenging for bulbar patients, these volitional tests, and so sometimes we have to fall back on oxygen saturation, capillary blood gases. Overnight studies are very helpful and the most useful, but quite challenging to organize. The 3-monthly visits are not very responsive to changes that might occur in between the 3-month period. They require individuals to come into the clinic and could be a burden on disabled patients, and obviously they were not pandemic compliant. The chart there is showing the various different respiratory parameters and how they decline steadily over disease course, but the ones that are mostly used, I would say, would be the SVC, the slow vital capacity, and possibly SNP. There aren't many centres routinely doing MIPS and MEPS. So will monitoring at home improve the situation, allow us to have information on patients' respiratory function in between clinical visits? And there are various sort of respiratory screeners, so they're not not actually full spirometers, but they do use algorithms and things to predict vital capacity and other measures. And there has been some work looking at the utility of these. So in the US, a study which was a proof of concept study simulated home telemedicine when actually the patient was just in the next room doing the video call, and they were correlating the measurements done in the room coached via a video call with those done in person, and they had a good correlation. And in Ireland, a similar correlation was seen. They were completing remote assessments, so they were at home, but they were coached through a video call to do the lung function tests. And people found it acceptable, and you see that 76% felt able to do the lung function tests without the video support. They would not feel able to be able to do it without the video support. So that's a concern. However, in the Netherlands, so this was a study where using Spirobank Smart, individuals had training at baseline, and then had a baseline measurement done, one which was supervised and one which was not supervised and not coached, and they had good correlation. And then they continued to do it on a monthly basis, and then at 3 months they had another dual measurement, a supervised one and then an unsupervised one, and again a good correlation. Individuals in this work found doing the monitoring quite easy at home, and so in this study it seemed as if it was feasible, so slightly contradictory to the Irish experience. And I think it depends how clinics and services are set up. Lessons from other diseases. So in COPD, remote and portable spirometers are used. There was a nice meta-analysis of this. Although largely these were remote spirometers, so more portable spirometers being used in primary care settings rather than individuals doing it by themselves, but showing that the readings were useful in the clinical diagnostic pathway. And then in perhaps more closer to ALS, in the Duchenne muscular dystrophy field, remote spirometry is considered to be reliable and is used both in care and clinical trials now quite routinely. So in terms of the clinical trials, if we just focus a little bit more on those, there are many clinical trials going on at the moment. The landscape is crowded, it's fair to say. There are a number of platforms going on in different countries, and then alongside that a number of commercial and academic-led standalone studies of single assets going on. So capacity to be able to deliver clinical trials with all the different studies that is going on is a challenge. And these next two slides just show the different existing and emerging therapies being investigated in many different pathways. but really all of them are going to need to use a functional outcome measure, and vital capacity is an outcome measure that is regularly used. And I showed the ADORE protocol, which is a pretty standard protocol, which was including pulmonary function as an outcome measure. Why is it used? It correlates with survival. It correlates with function, so we can see the correlation with ALS-FRS. It's an outcome measure which is important to patients and makes sense to patients, and it's accepted by regulatory bodies. The recent trials, so the Centaur trial of phenylbutyrate and TUDCA used SVC, and the VALOR study of from Biogen looking at tofersen, again using SVC as an important functional secondary outcome measure. A typical clinical trial protocol here, you don't need to read all of the detail there. What I'm trying to get across here is the complexity and the burden of these trials, and these are often lengthy visits, so patients are often there for many hours, and these are patients with significant disability. So when designing the protocols, I think it's also important to think of the timing within the day about when some of these things are being done. If the vital capacity is being left to the end of the visit, at the end of a 4 or 5 hour visit, and particularly if you're wanting to get reproducibility of the measure, then it needs to be brought forward, I think, earlier into the visit. This is from the Empower study, which was a Biogen-sponsored study a few years ago, dexpramipexole, and this is missing data. So this shows that the respiratory measure is a most likely measure to be missing. So nearly 40% missingness by the end of 12 months compared to functional rating score, which is a questionnaire with missingness below 5%. So why is that happening? It might be that as people have progressed, that developing bulbar dysfunction, and so it's more difficult to do the measure, so the measure is missing, or there may be other reasons. So I think what it's showing is they might not be coming to the centre, so some of those other things can be done remotely. So particularly the FRS and a blood sample can be done remotely. So designing trials with that in mind, and retention and reducing burden is important. And just because something's in a protocol doesn't mean that you're going to get it. So the pandemic, so Empower happened before the pandemic, so it's not the reason for the missingness there. But the pandemic did cause problems in terms of care and the way we delivered our trials. In terms of trials recruitment suspended, trial visits suspended, and outcome measures such as respiratory function were not possible, either because patients couldn't come to the trial centres, or in some circumstances it was classed as an aerosol generating procedure, and you weren't allowed to do spirometry. So it was a very challenging time. So what new approaches can we take? What can we use technology for to improve this situation? So in terms of care, it's about improving monitoring communication, reducing the burden. In terms of the trials, I think it's about improving trial data collection and reducing the burden of participation. So we've put together a roadmap for how digital health technology could be used in care and trials. So this is with the NCALS network and the TriCALS consortium within NCALS. So thinking about what it is we want to measure, it's important to patients, it's relevant to the treatments that we're giving patients or that we are evaluating, and minimizing burden. How are we measuring it? Thinking about the sampling frequencies, and we have some indication about what would be acceptable and what isn't acceptable. How we integrate all the data coming from these different technologies, and then thinking about the implementation from a regulatory perspective, and then in terms of care, about reimbursement. And so you could imagine, and we are developing something along these lines. So Paul Hardiman is leading on this Precision ALS initiative within Europe, where we're trying to bring the clinics, the registries, and eventually the clinical trials into one joined up network. And so you could imagine that we're taking remote data, actigraphy, FEC, weight speech. It's been reported via a mobile hub and coming into a database along with the clinic data that we have, any registry data that we have, and with trial data. And these are bidirectional, so that they may be pulling information into the database or pulling information out of the database with unique identifiers. So this is quite an undertaking, but it's something that we are keen to progress. And in terms of the devices and things we might use, I think one of the principles really would be to become agnostic of the device. So if we want to look at accelerometry, it wouldn't matter which device was being used, we'd be able to standardize the data and then feed it into the centralized portal to provide information for care or trials, and similarly with respiratory measures. So the issues with respiratory monitoring, when we think about whether it be how we might use technology, spirometry or a screener. So I think there's a difference clearly between what you do in a pulmonary function lab and these handheld devices that people are using at home. And so it's being aware of those differences and what you're trying to achieve and what you can do with them. Should the assessments be supervised and coached or unsupervised? Are they connected or are they patient reported? So are they Bluetooth connected? So something like the Spirobank Smart is Bluetooth connected, or is it more of a low tech device where the patient reads out the result and just uploads it themselves or rings it through? And in terms of clinical trials, are these exploratory? And at the moment, I think they are exploratory outcome measures. And hopefully in the future, when we've got better data of how they're performing in clinical trials, they'll be approved and recognized by the regulators. So I hope what I've tried to do today is to give an overview of the field of motor neuron disease and where we are in terms of particularly what standard care is and how that's changing, the clinical trial landscape and some of the challenges within the clinical trial landscape, how we might use technology going forward. And I've tried to put that through a lens of respiratory monitoring as one of the key important things, both for care and for trial outcomes. I think I'm hopeful about the future, and certainly over the next decade, I think we're going to transform the outlook for people with this disease. I think gene therapies are showing real promise for subtypes of motor neuron disease. There's been the huge advances in the understanding of our biology of the disease, and the assets that are coming forward into clinical trials now are exciting and targeting these new pathways. And I think in terms of trial design, we're getting things more efficient. We're improving our inclusion criteria and how we deliver these trials, perhaps using technology, using platform designs. I think we're going to be able to assess more effectively these exciting assets. I think that's the end of my presentation. Thank you for your attention.
Well, thank you very much, Chris, for that. Really appreciate it. It was an excellent presentation. So we've been looking at some of the questions that are coming in. And one thing you mentioned in terms of an outcome in clinical trials is obviously vital capacity measurement. So we've had a few questions around the considerations of using either slow vital capacity or forced vital capacity as an outcome measure in a clinical trial. I see noted a few of the studies were using forced vital capacity and some were using slow vital capacity. So just your thoughts on which is the better technique for an outcome measure in an ALS study.
So I think the field has evolved. So I think if you look back, so 10 years ago, most would have been using forced vital capacity, but now there's been a shift to a slow vital capacity, and I think the reason for that is it's easier for the patients to be more consistent with a slow vital capacity, and that's related to features of the disease. So the coordination that's required for the force maneuver is more difficult for people with upper motor neuron dysfunction and the coordination issues that causes. So I think patients find it easier and we get more consistent results from that.
Okay, great, thank you for that. And then there was another question specifically around supine measurements versus seated measurements, and the considerations for that, and how much do the values change between those two types of measurements, and whether that's useful within a study. So
in terms of when people are supine, you can get a drop, sort of should be below less than 10%. I think it varies in people. So a few percent drop when people becomes supine from sitting. If it drops more than 10%, that's a sign of significant diaphragmatic weakness. So again, it is important that it's protocolized appropriately how the measure is being done in the clinical trial, so you don't have trial sites, perhaps some doing them as a supine, some doing them as sitting. So there are differences. There can be differences. So to reduce the variance, it's important that it's protocolized.
Yeah, we just had another question just now, and again about lung function, Chris. It says, is there any evidence to suggest that lung function training can slow lung function decline in ALS patients?
No, not strong evidence. But it's an interesting question, and it's something. So we a few years ago did a study of cophasis, so mechanical insufflation, exsufflation, and in the control arm we used breath stacking, which you could think of as some form of lung training. And we were surprised. We were told we couldn't have, in the ethical committee, we were told we shouldn't have a placebo arm in the study. We were interested in the mechanical cough assist, and did it prolong survival or improve respiratory outcomes. So we put the breath stacking in because the ethical committee didn't want us to have a placebo arm, and they did just as well. And I don't believe it was because it made the cough assist was making no difference. I think because there was a benefit of the breath stacking and perhaps improving lung function. So I think there may be something in that, but it's not proven yet. But it might be something worth exploring further.
Okay, great, thanks for that. So I'll let you off the hook now. That was the last question that we had today. So I'm actually, I think at this point, we're going to finish off the webinar. I just want to thank everyone who's attended today. And just to let you know that, as I said before, that this webinar is recorded and will be available on our website. We'll also send out a link to this webinar so that you can download it and see it for other colleagues that may not have been able to join today. So lastly, Chris, I just really want to thank you again so much for your time today and for being available to talk to us on this topic. We really do appreciate that. And with that, I think I'd like to close the webinar and wish everyone a very pleasant day wherever they may be. And thank you again for attending. Cheers.
Thanks.